Listeria Monocytogenes. It is both saprophyte and commensal. It is a gram-positive rod. Talking about its motility, it is non-motile at 37 degrees Celsius, but at room temperature of 25 to 28 degrees Celsius, it is motile and it shows differential motility. The motility is tumbling or end-on-end motility. This is because of the eruption of peritrichous flagella around the bacteria at room temperature. It is non-capsulated. It is a facultative aerobe. It grows at wide temperature range from 4 to 45 degrees Celsius. It is a psychrotroph. That means it can survive at low temperatures. Therefore, cold enrichment is used to isolate it from others as they cannot tolerate such low temperature. Biochemical reactions. It is catalase positive and CAMP test is positive. Listeria monocytogenes, when it is inoculated perpendicular to the streak of Staph aureus on a blood agar, there is a formation of narrow zone of hemolysis. On a blood agar, Staph aureus is inoculated and perpendicular to it, Listeria monocytogenes is also inoculated. So there is hemolysis, narrow zone of hemolysis around Staph aureus and Listeria monocytogenes. This hemolysis occurs because of the toxins produced by these bacteria which lead to the lysis of the RBCs in the blood agar. And the area where these two bacteria are close, there is formation of shovel or wedge shaped area because of increased hemolysis because of the synergistic effect. Listeria monocytogenes produce hemolysin that is known as listeriolysin, which synergize with Staph aureus beta lysin, leading to increased hemolysis in that area. This is CAM test positive. Anton test. It is a diagnostic test for Listeria monocytogenes. Instilling suspension of Listeria into conjunctival sac of guinea pig leads to keratoconjunctivitis in guinea pig within 24 hours. It reflects its invasive property. Selective medium for Listeria monocytogenes is Palchem agar. Virulence factors and pathogenesis. The virulence factors include internalins A and B. These are the surface proteins present on the surface of Listeria which help to invade the epithelial cell. These interact with the E-cadherin on the host cell. E-cadherin are the cell adhesion molecules present on the surface of host epithelial cells. Internalin A binds to E-cadherin and this causes the entry of Listeria into the host cell. And mainly these include intestinal, placental or hepatocyte because E-cadherin is highly expressed in these cells. After entering into the host cell, it is engulfed by the phagosome or endosome, but it breaks out into the cytoplasm with the help of listeriolysin O. It is a pore forming toxin which is activated at the acidic pH inside the phagosome. Phospholipase A and B also help in breaking phagosomal membranes. Actin assembly inducing proteins known as ACTA hijacks host actin machinery, form actin comet tails to propel itself across the cytoplasm and into neighboring cells. So Listeria spread cell to cell directly, evading immune detection. So this is Listeria monocytogenes, and on the surface of the host cell is E. cadherin. It interacts with E. cadherin and moves inside the cell, where it's engulfed by the phagosome, but it causes lysis in the phagosomal membrane and moves out with the help of listeriolysin O and moves freely into the cytoplasm and replicate itself, and with the help of ACTA it moves across in the cytoplasm towards the cell membrane and moves into another cell. Epidemiology: it is a saprophyte present in soil, water, sewage, animal carriage, cow, sheep in their GIT, humans 0.51% as GI commensals. Mode of ingestion: milk, soft cheese, meat, refrigerated food. Most common serotype causing human GI disease is 1 by 2A and 1 by 2B. Most common serotype causing invasive disease is 4B. Listeriosis: in healthy individuals, febrile gastroenteritis with incubation period of 2 days. In elderly, immunocompromised, meningitis, meningioencephalitis. In pregnant women, there is 17 times risk of bacteremia that can lead to chorioamnionitis, spontaneous abortion, stillbirth. Transplacental infections cause granulomatous infantiseptica, characterized by skin pustules and multiple visceral abscesses in neonates. Perinatal transmission from infected birth canal can lead to neonatal septicemia and neonatal meningitis. The drug of choice for Listeria monocytogenes is ampicillin. For invasive disease, gentamicin is also included with ampicillin. If allergic to penicillin, cotrimoxazole.